So the correct incoming nucleotide is DTTP. And this was in the absence of magnesium. And then he incubated these crystals in magnesium for different amounts of time, froze the crystals, collected data, saw all the structures. And this is what the basic structure of this DNA polymerase 4 in complex with DNA, which is shown here, and incoming nucleotide, which is DTTP, shown here. So DNA polymerases always adopt this, structurally adopt this right-handed topology, and they will always have a palm domain, a fingers domain, and a thumb domain. And the active site residues are always in the palm domain. In case of polymerases like DNA polymerase 4, which is a member of the Y family of DNA polymerases, they have another domain called as the polymerase associated domain, which is very important for holding the DNA in the correct position. So this is your template DA, incoming DTTP, and these are the catalytic residues shown here. They are always acidic residues, usually aspartates or glutamates. So this is the electron density that you see here for the primer, terminal nucleotide of the primer and the incoming DNTP. And what Jitesh could look at this electron density and see that the bond formed between the 3' OH and the alpha phosphate over time, and he could identify some 15 different intermediates during this reaction. So I am not going, and the identification of these intermediates shed light on the chemical reaction that propels DNA synthesis by DNA polymerases. There are few things which got clarified during this study, but I am going to show here only the key, most important finding here. So this is the situation when the ions had just entered into the active site, and this is the situation later. So you can see that a bond has formed here between the 3' OH and the alpha phosphate. And what was surprising, what we saw was that this pyrophosphate moiety has actually split into two inorganic phosphate molecules, and one of them has diffused out. So it looks like the hydrolysis of the pyrophosphate is part of the DNA synthesis reaction, and we were able to confirm this using other biochemical assays also. So basically, to modify what you saw in the textbook, which was only till what the textbooks tell you basically is that you have pyrophosphate and then an accompanying enzyme called as pyrophosphatase. It hydrolyzes the pyrophosphate to phosphate. But here we see that the DNA polymerase itself can do this reaction, pyrophosphate to phosphate, without the need for pyrophosphate. Why is this important? Now, if you look at these textbooks, they'll tell you basically that one bond is broken and one bond is formed. So the free energy change that happens during the DNA synthesis reaction, that is close to zero. Okay, so when you have a free energy change close to zero, the forward reaction, that is synthesis of the bond, as well as the reverse reaction, which is breaking of the bond, will have equal probability. So why is it that polymerases, instead of synthesizing DNA, they don't degrade DNA? You never see them degrading DNA. And so the reason in the textbooks given is that pyrophosphatase, it will hydrolyze the pyrophosphate moiety, and together these two reactions will have a highly negative free energy change, and that will ensure that it always moves in the forward direction. But this question that do you carry out DNA synthesis routinely in labs while doing PCR? But nobody adds pyrophosphatase there. Without pyrophosphatase itself, you are getting a lot of DNA synthesis happening. And so So this was the puzzle that had been in the field for quite some time. And this is a snapshot of the idea report published by Karim Mullis in 1987 after he invented PCR. And here you can see neither in method 1 or 2 there is any pyrophosphatase involved. So basically what is happening is that you have the primer nucleotide 3'OH and the alpha, beta and gamma phosphates. This is the incoming DNTP. So initially this bond is formed, the phosphodiester bond is formed, you get a pentavalent interpingiate, and then this bond between the alpha phosphate and the bridging oxygen breaks. So you get the pyrophosphate moiety here. Now you can get a reversal of this entire reaction through a process known as pyrophosphorylasis. But it rarely happens because probably the DNA polymerases hydrolyze the PPI into 2 phosphates. And till here, the delta G of the reaction is very close to 0. The free energy change is very close to 0. But once you hydrolyze the phosphate, then the free energy change becomes very highly negative. So you will never get this reverse reaction. So the chances of this reverse reaction happening are very, very low. And that is how you ensure that DNA synthesis by DNA polymerases is always moving in the forward direction. So this is one of the important findings from the lab in the case of DNA polymerases. And this paper was published as a breakthrough study in nucleic acids research. And recently another lab working on another polymerase also saw that the pyrophosphate moiety gets hydrolyzed in the DNA polymerase active site. They saw that in case of Rev1. Okay, so one of the other things that I was going to talk about is this molecule. So this is RCTP, and it is different from DCTP that it has a hydroxyl group at 2' position. So the DNA polymerase has to ensure that it selects this and not this, because Rntps generally are more susceptible to cleavage. So the presence of Rntps in DNA can lead to genetic instability. And in case of prokaryotes, it's very well known that if Rntp incorporation happens, then the organism just dies. In case of to diseases like cancer. So the polymerase has to be able to select only DNTPs and not select Rntps during DNA synthesis. And this is again what is written in your textbooks as that discrimination is mediated by steric exclusion of Rntps from the DNA polymerase active site. So how does this steric exclusion happen? So this is the active site of DNA polymerase 4, okay, which is very good at preventing ribonucleotide incorporation. And what you see here is that you have this sugar moiety here, the ribose sugar. It stacks over this aromatic residue side chain, the phenylalanine side chain, the aromatic ring of phenylalanine. And this position is usually occupied by either phenylalanine or tyrosine, which are both aromatic residues. So this stacking is there, and if there was a 2' OH here, it would clash with the pi electron cloud which is present over the aromatic ring. And therefore the RCTP will not be able to sit here properly. And that is how they are excluded from being incorporated. So this is what you will find in the textbooks. So again, you can see that if there is a 2' OH here, it will clash with the pi electron cloud over the aromatic ring of this phenylalanine residue. Now, Mary, who was a student in the lab, she was working on this DNA polymerase from Mycobacterium magmatis. And what she saw was, so that DNA polymerase naturally does not have the aromatic residue. It has a leucine there instead of a tyrosine or a phenylalanine. So one would expect, okay, this polymerase should be able to incorporate ribonucleotides more than DNA polymerase 4. And that was correct. It was incorporating a lot of ribonucleotides. So Mary changed that leucine to tyrosine L14Y. And surprisingly, it is still able to incorporate a lot of ribonucleotide. So as compared to DNA polymerase 4, no incorporation at all. This is very stringent exclusion of. So what she thought that there must be something more in DNA polymerase 4, which other than the steric filter that prevents ribonucleotide incorporation. So she solved structures of MSDPO4 in complex with DNA and incoming nucleotide, and in complex in complex with either DCTP or RCTP, and she could see that both of them were sitting happily in the active site. There's no difference in the structure. That is why this is getting incorporated without any problem. Now, when you engineer in that steric filter, instead of leucine, if you have tyrosine here, still it is able to sit, the RCTP is able to sit in conformation. This is the 2' OH here. So this is a RCTP and this is DCTP. This is the 2' OH here. So even if you have RCTP, it is able to sit in the active site without any problems, even if you have engineered the steric filter. Now, no, these are all X-ray structures. That is no, that is all crystal structures of the ternary complex. There is no modeling in this one. Okay. Okay. So when you compare all these structures, what you see is that when you have the steric filter, this RCTP, the sugar and base, they are able to move a little, to avoid that steric clash and still be in a conformation that is compatible with productive catalysis. But that was definitely not happening in case of Pol4, DNA polymerase 4. So she compared the two structures, the complex of DNA polymerase 4 with DNA and incoming, and that of MSDPO4 with DNA incoming nucleotide, to see what was the difference. So what she saw was that there is a threonine residue in case of DNA polymerase 4, which forms interactions with the incoming nucleotide. And in case of MSDPO4, that is that the equivalent residue at that position is cysteine. So cysteine won't be able to form these interactions. And so this gave rise to the hypothesis that there is a polar residue present at a particular location in DNA polymerases, which can interact with the incoming nucleotide and prevent ribonucleotide integration. And then she did a lot of biochemical experiments, mutated the cysteine 47 residue to threonine, and then it was not able to incorporate any ribonucleotides. So while the enzyme is not able to incorporate ribonucleotides. And since if ribonucleotide incorporation occurs, it is lethal for the bacterium. So if she overexpresses MSDPO4 in E. coli cells die. But if she overexpressed C47T mutant of MSDPO4, they were able to survive quite a bit. So basically, this what tells you that in addition to the steric filter that you see in textbooks, there is also a polar filter. And the way it works is that it will interact with the incoming nucleotide and pull it closer to the enzyme surface. And if it has a 2' OH, that 2' OH will clash with the enzyme surface, as you see here. If it doesn't have a 2' OH, there is no steric clash seen here. And that is how it prevents ribonucleotide incorporation. Okay. So in case of ribonucleotide incorporation, another thing that Shilpi Nagpal, who's a student in the lab, had shown was for this enzyme POLX from Staphylococcus aureus. So POLX is a X family polymerase, and bacterial X family polymerases always have a proofreading domain. It is very different from the usual proofreading domains that you see, but it has the same function that it can carry out. It has a 3' to 5' exonuclease activity, which can remove any misincorporated nucleotides. So if Watson-Crick, if nucleotide incorporation according to Watson-Crick base pair rules, then the exonuclease jumps in and removes those removes a few nucleotides, including the wrong nucleotide from the primer. But what in the case of misincorporation of Rntps, what this was all by doing biochemical assays with wild type enzyme and with mutants. What she saw was that the proofreading domain, if Rntps are incorporated by mistake, then the proofreading region of the POLX enzyme can remove Rntps also. Even though the Watson-Crick base pairing rules might be satisfied, but if it is a Rntp, the proofreading activity can remove that also. This is how the proofreading activity can contribute to preventing by removing Rntps. And similarly, there is this molecule known as 8-oxo-DGTP. So it is a modified DGTP with a oxygen atom at this 8 position here. And this is the main, whenever there is reactive oxygen species generated in the cell, it can go and oxidize the nucleotide pool. And this is by far the main product of major oxidized DNTP in the nucleotide pool due to ROS. So the problem with this oxidized 8-oxo-DGTP is that it will get mispaired opposite template DA instead of template DC. And that happens because it forms this Hookstein base pair instead of a Watson-Crick base pair. So the base of the modified nucleotide, it flips around this glycosidic bond from what is known as anti-conformation to this syn conformation, and it forms hydrogen bonds like this. So you get a Hookstein base pair like this. And the distance, this is the C1' atom here, this is the C1' atom here. The distance between these two is usually in Watson-Crick base pairs is usually around 10.7 angstrom. And in case of a Hookstein base pair like this, it is the same distance. So polymerases can get fooled to incorporate this opposite A, to a significant amount, and transversion mutations in the genome and can be lethal. And mitochondrial DNA polymerase, even low amounts of 8-oxo-DGTP incorporation is very bad for the cell. And this is a problem especially for the malaria parasite. Malaria parasite is special because it has 3 genomes. It has a nuclear genome, mitochondrial genome, and then it has this organelle known as the apicoplast, which has its own circular genome, about 35 kb, which is very AT rich. And inside the apicoplast, there are lots of biochemical pathways which are going on, which give rise to a substantial amount of ROS. So this genome is very susceptible. So the nucleotide pool will definitely get oxidized and have large quantities of 8-oxo-DGTP. And because this genome is AT rich, there is very high chance of 8-oxo-DGTP incorporation while replication of this genome. This genome is basically replicated by a special enzyme known as PF-PREX. And PF-PREX is nuclear encoded. It starts out which has polymerase, helicase, and primase activities. And then once it enters the apicoplast, it gets cleaved into 3 different proteins. So the polymerase module we call as PFP-Pol. And we wanted to see what would happen regarding PFP-Pol DNA synthesis activity in the presence of 8-oxo-DGTP. So what Meenakshi was able to show is that the polymerase enzyme does add 8-oxo-DGTP opposite DA on DNA, but it has a proofreading exonuclease domain which can remove these wrongly added 8-oxo-DGTPs from the primer. And can do this because in the exonuclease domain there is this specific motif QQNS, which the residues of which forms interactions with the damaged nucleotide. And this QQNS was only seen in this Plasmodium enzyme and not in any other A family polymerases. So it has acquired this capacity to be able to bind DNA which has a terminal 8-oxo-DGMP and so ensure that it can remove it. And so this enzyme has acquired this ability to be able to prevent the deleterious effects of ROS on nuclear DNA synthesis in the apicoplast organelle of the malaria parasite. So this is a schematic of the study. Here you have DNA polymerase 1 from E. coli, which is meaning the famous Kleno fragment comes from this enzyme. And this is the closest structural homologue to the PF-PREX polymerase from Plasmodium falciparum. So you see that both of them incorporate 8-oxo-DGTP opposite A, but whereas DNA polymerase 1 just continues synthesizing so that that mismatch is retained. Here in case of PF-PREX, the exonuclease domain carries out editing of the newly synthesized DNA so that this misincorporated 8-oxo-DG is removed, and then after that it can resynthesize again to ensure that there is no mutation. So in this way it protects the highly AT-rich genome of the apicoplast from the deleterious effects of reactive oxygen species. Now, so this is a little of an aside from the general studies that I've shown you in the lab. Again, you have this 8-oxo-DGTP, and what was that DNA polymerase 4 can incorporate 8-oxo-DGTP into the genome, and that is lethal for the cell because once it starts incorporating 8-oxo-DGTP in the cell opposite DA, repair pathways are activated and when these repair pathways are activated, they lead to the presence of double strand breaks in the genome, which is generally very lethal for the, and there are more number of double strand breaks than the cell can repair, that will lead to death of the cell. So Jitesh had seen the same thing, and what there is substantial. So these are kcat by km values which provide the catalytic efficiency for these incorporations. So the incorporation of 8-oxo DGTP opposite DA is about there's a significant amount of incorporation possible by DNA polymerase 4. So he saw the structure, and you see in the structure that there is a Hookstein base pair compared to the Watson-Crick base pair. And what he saw was that these residues S42 and S55, they form interactions with the modified base to hold it in this syn conformation so that Hookstein base pairing can occur. So he mutated these two residues to ALA, and he saw that the ability to incorporate 8-oxo DGTP is decreased substantially for the mutant protein. Now, so what was the use of doing all this? So what he used it as a tool to resolve a very important question that was, there was a controversy regarding how antibiotics act. So there was a paper which said that ROS, the presence of antibiotics give rise to ROS, and that contributes substantially to the lethality that appears due to antibiotics. But there were other papers which didn't agree with this observation. So what we were able to do is that, this is the hypothesis that when you treat bacteria with antibiotics, the reactive oxygen species appear. They give rise to 8-oxo DGTP. This 8-oxo DGTP is incorporated into the genome by Pol4, and that ultimately leads to double strand breaks in the genome, and that leads to death of the cell. So how to check this? So he did growth assays where he grew bacteria, the bacteria which where the Pol4 gene is deleted. You transform them with plasmids bearing wild type or mutant genes of Pol4, and then you grow them in the presence of H2O2. So he saw that when you have wild type, there is substantial, percentage of survival is low. But if you have if you mutate those two residues which are important for that Hookstein base pairing, then the survival is higher. And now, if you so in the presence of H2O2, the activity of Pol4, the 8-oxo DGTP incorporation activity of Pol4 contributes substantially to the cellular lethality. Now he did the same thing. So he grew it in the presence of limited amounts of norfloxacin, which is a fluoroquinolone antibiotic, and there also he saw the same thing. So basically, this is a proof that when you treat cells with antibiotics, ROS are generated and also contribute substantially to the lethality. Support that initial report, and there are other papers also which support that initial report. Did for bacterial cells. So no, at least in this context here, the polymerase is very important for that ROS, the lethality due to ROS to manifest itself. So this is a summary. You have ROS. When you have bacterial antibiotics, they have cellular targets which lead to death of the bacterial cells, but also when you treat bacterial cells with ROS, with antibiotics, they give rise to ROS, which oxidizes the nucleotide pool, and you have 8-oxo DGTP molecule generated, which are incorporated by Pol4 into genomic DNA that leads to double-stranded breaks and lot of mutation, which can also contribute to the death of the organism. So here we were able to use DNA polymerases as a tool to study an important aspect of antibiotic action. And so this is what DNA polymerase is, meaning there are lots of papers. So the gene which encodes DNA polymerase 4 for in E. coli is known as DINB. And DINB, if you put DINB in PubMed, there will be at least 200 papers on DINB. And a substantial number of them are about the role of DINB in adaptive mutagenesis, that it has a mutator activity. And basically what happens is you have, when you have stress, then DINB is upregulated or Pol4 is upregulated, and that gives rise to different kinds of mutations in the genome. Okay, so you can have different cells with different mutations. In a cellular pool, you will have different cells with distinct mutations in them. And that is giving rise to multiple genotypes which can be subjected to natural selection. So there is a chance that one of these cells will have that golden set of mutations which can neutralize the stress that is present in the environment and help it survive. So all the other cells will die out but survive, and then it can replicate, it can proliferate and colonize. And this kind of phenomena, adaptive mutagenesis, is implicated in the effect of drug resistance in case of pathogenic bacteria. So Pol4 plays a very important role in this. And we wanted to figure out what is the molecular mechanism used by Pol4 to promote mutations. So the structure, it showed that there is this serine 42 residue which is involved in forming interactions with the incoming nucleotide, the template nucleotide, or both. So it helps hold the base pair in the active site. In all 4 cases, there are interactions between the serine 42 residue with the template incoming nucleotide or both. And if you mutate this residue, then the mismatches that you see, which are incorporated by Pol4, they reduce substantially. So the presence of this residue contributes substantially to greater activity of Pol4. And this is, if you grow cells in the presence of rifampicin, then wild type you see a lot of colonies, but if you have a S42A mutant, then the number of colonies decrease substantially. So this was a structure solved where you have AC mismatch. There is no hydrogen bonding possible between, in case of adenine and cytosine bases, unless this nitrogen is protonated. So in spite of there being no hydrogen bonds here between the two, still C is able to sit opposite A in the active site because this serine can hold it there in a conformation that is compatible with enzyme activity. And the serine residue, it is present at a strategic locations towards the minor groove, and it holds the incipient base pair in a even for a mismatch in a conformation compatible with productive catalysis. And this design apparently may be important. If you have too many mutations, it is going to be bad for the organism because genetic viability will go down. If you have too few mutations, that is, then you're not generating enough genotypes for natural selection. So maybe this putting a serine residue towards the minor groove of the incipient base pair is a good way of generating enough mutations in so that in calibrating the mutator activity so that you generate enough genotypes without losing genomic viability. Okay. So the last section is based on translesion DNA synthesis, which is another important function. You know, all of you are aware that the DNA has the blueprint of life and it has to be replicated. But what not many people are aware is that there are a lot of this DNA is subjected to a lot of chemical stress, both by endogenous agents, which are generated naturally during physiological processes like acrolein, or by exogenous agents like radiation or many chemicals, things like that. So because of these endogenous and exogenous agents, you get DNA lesions, which are modified nucleotides in the genome. This is the most famous of them, cis in TT base pair, cis in TT dimer, which is formed due to UV light. Or you can lose the base and you get a basic site, or you can get an exocyclic ring which on the adenine base. So all of these will prevent productive Watson-Crick base pairing, and therefore their presence in the genome leads to stalling of the replication fork. You have high fidelity DNA polymerases which will stall because they are not able to accommodate these enzymes in the active, these in the active site. So to rescue replication, you have lesion bypass polymerases which will step in, and they will be able to add nucleotides opposite the normal replication times come back. So this is known as translesion DNA synthesis, replication stalled at DNA lesions. And we studied this in the context of this lesion known as N2-furfuryl deoxyguanosine, which arises due to the action of this nitrofurazone antibiotic. So this is antibiotic. It is still used, in these hospital catheters. The inside of those catheters are lined with this kind of antibiotic, and it is also used sometimes for treatment. So the paper that came out of Graham Walker's lab in MIT, he showed that when this DINB gene is deleted, then the sensitivity towards this antibiotic increases drastically of E. coli. And so we wanted to figure out how is that this DNA polymerase 4 is reducing the sensitivity of nitrofurazone. And the surprising thing that he showed in this paper, and also we saw subsequently again, is that the catalytic efficiency for incorporation of DCTP opposite DG is lower than that for incorporation of DCTP opposite this furfuryl adduct. And this is very unusual for a translesion DNA polymerases. Meaning many organisms have translesion DNA polymerases. Humans have at least 4 of them. And for all of them, compared to undamaged DNA, the catalytic efficiency is always 10 to 100 times lower for damaged DNA. But here it is the exact opposite. It is better for damaged DNA rather than undamaged. What is the reason? So Jitesh did a lot of structures and biochemistry, but the gist is this. The DNA polymerase 4 molecule is such that you have these domains, the fingers, palm, and the polymerase associated domain. There is a thumb domain also, but that has been removed in this picture to show clearly what is happening. So the relative orientation of the domains are such that these gaps are formed between the domains so that the adduct which is formed can be present in them without sterically clashing with the enzyme atoms. So he solved 3 structures where the adduct is present at different positions during DNA synthesis. So this is the incipient base pair when it is present in the active site. Here you can see that there is space here that adduct is present over here and it will not the enzyme. After one round of DNA synthesis, it will be present here. Again, that minor hanging in a space and it is not clashing with any of the enzyme atoms. After 4 rounds of synthesis, it is present over here. Still there is space here. So this what you see here is very different from what you would see in case of high fidelity enzymes, where there is a very snug fit, and if there is something hanging off from the minor groove, it will clash with the enzyme atoms. So that is why the high fidelity enzymes like DNA polymerase 3 or even DNA polymerase 1, they won't be able to synthesize DNA when this lesion is present. And recently Meenakshi has shown that for the PFP-Pol enzyme, which is the polymerase module of the PFPrex enzyme, which is the apicoplast genome duplicating enzyme in Plasmodium, that also, in spite of in spite of being a family polymerase, which are generally high fidelity enzyme, it has the capacity to carry out translesion DNA synthesis. And it can do that because there are residues present in its active site which are unique to PFP-Pol, which can hold that damaged nucleotide properly in the active site. So this is some of the things that I wanted to show during this talk. And the future directions for the lab is that we would like to start studying functionally important complexes of these polymerases with their partner proteins, or for these microbial DNA polymerases, or for viral RNA dependent RNA polymerases with their partner proteins using crystallography, small angle X-ray scattering, and cryo-electron microscopy. And also try and carry out structure based drug discovery to identify potential inhibitors of microbial DNA polymerases and of the viral RNA dependent RNA polymerases. So to see if we can find some molecules which can be used for therapy. So with that, these are the current members of the lab, and the work that I showed was done by these past members. And I would like to thank the Central Instrumentation Facilities and the ATPC, and the collaborator on the Furfulir project who made those oligonucleotides with the modified nucleotide, who's at IT Bombay. Since this work started in NCBS, I'd like to thank the funding agencies, NCPS, TFR, DAE, and then RCB, DBT, and SCRB, who have funded this work. For X-rays, we use the X-ray diffraction facility at RCB, and also the ESRF access program, which is funded by DBT, has to be acknowledged, because without this program, it would have been very difficult for us to access synchrotron sources, which are very, very important for the work that we do in the lab. For the work shown here, ESRF data was used, but we have also collected data at another synchrotron called as Electra, and that program is funded by DST. Okay, thank you. Who goes first? Yeah, there is one more. So in case of the for about Mycobacterium tuberculosis, usually the bacterial chromosome is indicated by a Mycobacteria, which is very special, it has a DNA E2 also, which is a error prone polymerase. That is rise to drug resistance. But these enzymes may also play roles. With polymerases, it is very difficult to do these knockout like experiments because there is always redundancy. And you can have mouse knockouts of because there will be another polymerase which does their function. But it has been clear DNA E2 is responsible for these antibiotic resistance that are case of Mycobacterium tuberculosis, but these other enzymes may also play roles in that. Yeah. It helps mutator activity. No, what? Yeah. So serine 42, its role is that normally you will get Watson-Crick base pairing happening, and that polymerase will synthesize normal DNA. But because that serine is present, it can also at some low frequency add mismatches. So usually there is especially true for B and Y family polymerases. So I didn't show the data here, but for like polymerase, DNA polymerase 2, E. coli has 5 polymerases, 1, 2, 3, 4, 5. 2 is a B family polymerase. And we had done experiments with 2 also where the residue, polar residue was mutated and it acquired the ability, there was substantial increase in the ability to add Rntps then. So this strategy is there in at least in Y family and B family polymerases. And then it becomes very important because B family polymerases, in case of eukaryotes, the delta and epsilon, which replicate the chromosome, are B family polymerases. So there are residue equivalents in these polymerases, which may play the role of the polar filter. And we also looked at these mutations associated with cancer in case of human B family polymerases. And we did find some possibilities, like I forget which one, but in one of them, that polar filter may be mutated, was mutated to something else, and it is possible that that leads to higher Rntp incorporation, which leads to genomic instability, which leads to cancer. But somebody has to test. So the short answer to your question is that that kind of residue is conserved in Y family and B family polymerases at least. Yes, yes. Pyrophosphate. What I would think was happening would be that it has this ability to intrinsically hydrolyze, but some amount of pyrophosphate is generated, and that can be hydrolyzed by a accompanying enzyme. So it could be that this is the primary mechanism and the pyrophosphatase is a backup. So pyrophosphatases are there, and in case of bacteria, Mm. No, but DNA polymerases are very, what you call they have very high rates of synthesis. Like, in case of DNA polymerase 3, which synthesizes the bacterial chromosome, when it is going full tilt, it is 900 to 1000 nucleotides per second, which is pretty amazing rate. So DNA polymerases are generally very fast enzymes. What? Everything from our lab only. Yeah. No, those are three different structures actually. Oh, they are not. There is no. No. This is not. No. If the rates are so high, no. Then it would be possible. So there's a. So that is how you get snapshots of what is happening as the DNA keeps synthesizing. As a. No, those are very different. Yeah. So basically, that there is some chemical moiety hanging off from the minor group, because they are always present in these gaps and channels which are generated by the relative orientation of the three domain. So it just zips through that. It's like if you have a railway line, and if there is something stuck on the rail, then the engine can't go past it. But if you have an engine which has like a gap throughout its body, so that it can just go over that thing which is stuck there. It's like that. So it will just go without going. All questions are intelligent. Protein. No. Meaning, here in this case, the polymerase and DNA is sufficient. Usually, like I showed, application 4. Usually, polymerases are bound loose, meaning not loosely, but tethered to this processivity clamp which encircles DNA. And they have this tool belt model where the bacterial clamp has two binding sites. So it can maybe hold replicative high fidelity polymerase on one and the translesion DNA polymerase on the other. And so when the replication fork is moving, it gets stuck at some damaged nucleotide. The replicative polymerase fork comes off and the translesion one comes in. So that is the amount of interaction with the partner proteins. Okay. Thank you. What? Okay. So there was a mistake. There was a mistake on one of the slides.